I'm Michelle, a postdoc in Tony's lab, and today it's my pleasure to share with you a little bit of my work. It's very interesting but challenging. What do we know about the brains in the children with deletion? In our daily life, tons of information we need to process and in our brains every second, even without our notice. Just imagine that there is a non-stop running supercomputer just sitting inside your head. You will admire your brain. It's the most complicated, sophisticated computational information process system have ever made in the world. It's such a difficult work for everybody, particularly for the children with deletions. You may have the experience like that. Sometimes you are wondering why kids are very easy to get bored and very easy to get upset in the school. But sometimes if they are doing their favorite games and really engaging it, it might be your time to get mad and be upset. You may ask why. One really good way to think about it is to understand their brain. Individual brains are different, unique, just like our fingerprints. It's so complicated and unexplored. Researchers will spend a lot of time but still barely understand it. But we do understand a little bit of these differences in the children of deletions. Let's take a look at it. First of all, some brain structures are either larger or smaller in the children with deletions compared to their similar age, typical developing peers. It's a review table recently published and only listed the most consistent findings from at least 2 independent research groups. So, some of the structures in the children with deletions are different from the typical developing kids. Not just the size, but the connection in the brains are also atypical, particularly in the specific areas at the back of the brain in these kids. Also, other features in the brain, for instance, the thinner cortex in these kids, and also reduce the complexity of the patterns on the surface of the brain. It's a long list and it can run on and on, but if we take a second look at it, we will notice that most of the atypical structures are just located in the middle of the brain. Plus, and very important structures we call hippocampus. What hippocampus is? Hippocampus is a very important structure located in a specific areas called the limbic system. It was reported that involved in the memory, spatial processing, and emotional process. And the size of the hippocampus has been reported that linked to general intelligence levels of the children, particularly in boys. So, on the other hand, the functional connectivities from hippocampus connected to the front of the brain has been reported impaired in the 22q deletion mice model. How about the structure? So far, there is no studies reported what the structural connectivities from hippocampus to the other part of the brain. So the primary output connections from hippocampus is the white matter fibers we call Fornix. Fornix is a C-shaped fibers traveling from hippocampus to the other part of the brain. So here is the questions. How can we measure it? How can we see it in the brain? Okay. An advanced magical technique called diffusion tensor imaging gives us the chance to study the connections inside the brains without opening it. So, similar to other organs, our brain contains a lot of water molecules, right? The water molecules travel all the time in different directions. So, DTI, for short, diffusion tensor imaging, give us the chance to use these water molecules as a vehicle in our brain to see and study the inside connections. Let's take a look at it, how it works. Here is a glass of water, and the water molecules inside this glass can travel freely in all the directions because there is no restrictions in it, right? How about we putting into some drinking straws? This time in this situation, the water molecules are trapped and they can only travel along the drinking straws here. So they are directional movements. How about more? If a lot of drinking straws in the brain, and that in this situation, the movements of the water molecules can form a liquid float, and it's visible and can be detected by the diffusion tensor imaging, and used this way, we can quote-unquote see the chat. This is just drinking straws, but keeping in mind, I'll show you how it works in the brain in a second. Similar to the drinking straws, inside the brain, the white matter fibers are detected by the diffusion tensor imagings, indirectly taking the advantage of the water movement. So here, this is a typical brain, a brain from our typical kids. In the D-team, I mapped to distinguish the direction. Fibers collected from the front to the back are colors in green, when fibers traveling from the top to the bottoms, this direction, the vertical ones, colors in blue. And the left and right are colors in red. So you see here is the corpus callosum Tony just mentioned is connecting to the brains left and right, so it's in red, right? It's blue chat up and down. Using DTI, now we can study the brain. Okay, let's go back to our Fornix story. Here, the first question we would like to ask is, can we find the Fornix in individual brain? If so, does it difference in the kids with deletion? So, to define and find the Fornix, we need to first define an area we should look at it. It's similar to if you search a place in a Google map, you need to define your current location and what's your destination and ask the Google map to generate the directions for you, right? Similar in the Fornix, what we did is at the front of the brain, we draw a place, we call it draw a seat. As accurate as possible, we did it in the individual brain. So that's the back. At the back of the front of the brain, we also draw another seat. And that we define your location A, location B, and what's the next? It's asked for the connection. So here, we use a really powerful software to ask for all the possible connections between these two seats. We ask the programs to run it 100,000 times because we don't miss any possible one, right? After that, we pick the top 1%, the most likely ones to get a really clean Fornix. Therefore, here we finally get a clean Fornix chat in the individual brain. We did this from brain to brain because we know every brain is different. Like our fingerprints, you cannot do it as a group. So, what we have here is some examples of what it looks like of the Fornix. The first row shows that if we ask all the fibers generated from the front seat, which is location 8, this is where you get the blue little thing in the brain. And if we ask for the fibers from the back seat, it looks like this. How about ask for the connections? It's the pink one. The pink color here represents most typical looking Fornix, and it's the other fibers nearby. The neighbor fibers are colors in either green or yellow, but it's not a typical Fornix. You may notice that in some brains, like this, the Fornix are perfectly connected. We categorize it as type 1 because it's typically connected Fornix. But in some of the brains, the fornix are connected in a really atypical way. It's less typical because it looks like it tangled with other neighbor fibers and not as clean as the type 1, right? Understand? Yeah. So we would ask the questions. How the fornix looks like in the typical developing kids and the kids with deletions. In total of 38 typical kids, most of them looks like have the really typical fornix. It's perfectly connected. How about the kids with deletion? Let's take a look at it. In total of 45 kids, less than 50% have the perfectly typically connected fornix. Most of them have a really unusual way. So we would like to ask the second question is why these atypical patterns more often shows up in the kids with deletions. One explanation might be the fornix in their brain may be less organized. Therefore, it's easier to tangle up with other fibers around. But how can we measure it? How can we know if it's well organized or not? So here, one approach we did is to pick out the only typical looking fornix, which is the pink section here, the pink one, and that try to study the integrity, how well it organized. How to measure it? Let's go back to our drinking straw example for the moment. In these two pictures, which one is better organized? Left or the right? Left, right? Yes. So since the water molecules are mainly travels from bottom to the top in this example, we can quantify these main directions with using a specific term. We call it fractional anisotropic, FA for short. You can ignore it. You just remember FA is representing the main water movement direction is from this. And another way is to use the complement index. We call it radial diffusivity. It's talking about the other direction. So use these two terms, we can try to quantify and summarize how well organized the fornix chats are in the brain. So go back to these examples. Higher FA will be in the left picture, right? And higher RD in the right-hand side, right? Yeah. So that's the fornix. As a group, higher FA and lower RDs are shown up in the typical developing kids as a group. And it highly varies between the kids in the kids with deletions. We can see some kids are just as typical as the typical developing kids, but some of them has extremely low F8. If we separate it further by the connection types we just talked about, the type A, the type 1 is the perfectly connecting fornix, and the type 2 is tangled with other neighbor fibers. We will notice that not just in the kids with deletion, but in the typical developing kids, the type 2 has lower FA compared to the type 1. So what does it mean? It means that in the kids with deletions, particularly in the type 2, it's less organized fornix fiber. You may notice that these less organized fornix fiber are not everywhere. It may happen in a specific areas. So the third question we would like to ask here is these less organizations across the entire chat, or is only focused on the specific areas, like this example, right? Because the bottom part is still well organized. It's just the top ones is dispersed a little bit. So again, what can we do? What can we, if we can zoom in and examine the entire chat to see which specific locations are less well organized, and we may understand the connections better. One way to do it is we can walk along the chat the Fornix from the front to the back, and that we may have the solution. So here we go. We try to cut this entire chart into different pieces. Here we choose 45 is because the length of the median in the group is 45. In that case, each piece represented 1 millimeter long of the Fornix chart. So now we can examine the charts in details. Here is the left FA, right FA, left RD, and right RD. Here is the front of the Fornix, the back. When we walk along the track, we may notice that as a group, the mean of the typical kids are higher compared to the mean of the children with deletions. But here, each of the thin lines are one individuals, and we can easily see there is a big variations from kids to kids. Even in the kids with deletions, some of them has extremely low FA, but some of them are just as high as the typicals. You can very easy to see here. The same thing happens in the RD if you still remember the example. And specific locations, yes. It's not a surprise. The primary differences between the typicals and the children with deletions are located at the back of the chart, where is the place right connecting to the hippocampus, particularly on the right side. In addition, there is a small chunk of areas at the very front end. Here, if you still remember, it fits in our very first slides. It's most of the atypical structures reported in the children with deletions are right in the middle of the brain. Because the Fornix is traveling like an arch in the middle of the brain, it's very reasonable to hypothesize that it may be influenced by the other atypical structures. Let's take a look at it. The middle of the brain structures, we call it midline structures in the imaging study. As Tony showed previously here, Elliott's previous work shows that the higher the incidency of extreme CSP is higher in the children with deletions. So where's the Fornix? It's here. You can see that it travels in the middle of the brain. How about if you have an extreme big CSP? It may have been affected, the structures of the Fornix, how it forms together, and higher FA or lower FA or higher RD, sorry. So, with a really big group of kids, which is close to 80 children with deletions, we can perfectly replicate our previous findings. About a quarter of the children with deletions have the extreme CSP, particularly in boys. And here you can see it highly varies within the group. Another important features in the midline structures are the ventricles. Collaborated with Dr. Owen Carmichael's group, we measured the size of the ventricles, which is here, the hole in the middle of the brain. So we find that in a really large group of kids, the children with deletions have bigger ventricles, particularly in boys, if we separate it by gender. And moreover, the size of the ventricles increase when they are older, particularly in boys. Again, here we are very easy to see the variations within the group. So far, we've already talked about several brain features, brain differences in the children with deletions, including the atypical brain connectivities and the atypical structures. So I would like to give you a really brief summaries to put all the pieces of information together. First of all, the main finding here is the hippocampal connections in the children with deletions are very unusual. They have the less organized Fornix, particularly at the regions really close to the hippocampus. And that it highly varies between the children with deletion. And then second, the brain structures in the middle of the brain, the midline, are really unusual. They have bigger CSP, they have bigger ventricle, and again, it's highly varied. And this one may, the midline structures may influence the hippocampus connection, which is the fornix, as you already seen in the picture. Very important message here I would like to bring up is we haven't found any of these single components above have any strong functional correlations, implications. But each of them have been commonly reported in the patient of schizophrenia. What does it mean? You may ask, does it matter? Is it the brain really matter? It's some really important questions, but unfortunately, we haven't have the really clear answer now. I would like to say, it depends. Yeah, brain is like a non-stop running supercomputer. If you agree, whether it functions well or not really depends on a lot of complicated factors in the real world. And it depends on what and how the hidden equations really work to generate a better outcome. It's a very challenging question waiting for us to answer. And I hope the following talks given by our other lab members will give you useful insight. Thank you very much, particularly to all our participants and their families.